Antimicrobial susceptibility testing. Antimicrobial susceptibility testing is performed on bacteria and fungi isolated from clinical specimens to determine which antimicrobial agents might be effective in treating infections. Need to distinguish pathogens from commensals. So first in the diagnostic process, so we need to identify what the pathogens are. And then once we have isolated the pathogens, we determine resistance of those pathogens. We do not want to determine resistance of commensals. We don't want to kill off our normal bacteria that live inside our body or the bacteria that aren't causing the problem. We want to kill off the pathogens, so it's very important to distinguish. Susceptibility testing, generally disc diffusion or dilution. Standards are published by Clinical and Laboratory and Standards Institution, CLSI, Worldwide International Organization for Standardization, ISO, standards are used. Diffusion is also known as minimal inhibitory concentration, or MIC. Minimal inhibitory concentration is the minimum amount of the drug that will still be effective in killing off that organism. When we treat patients with drugs, we don't want to give them high doses of chemicals. We don't want to give them high doses of drugs. So if we know the minimum inhibitory concentration, it allows us to give them lower doses of drugs that are still effective. Reasons and indications for performing antimicrobial susceptibility tests. So we should not perform antibiotic susceptibility tests on every single thing that comes down to the lab. There's reasons why we should and should not perform antimicrobial susceptibility testing. First and foremost is cost. We don't want to put unnecessary costs on the patient, but also when there's probable cause to suspect the isolate is involved in the patient infection, then we're more likely to want to do susceptibility testing. Like I mentioned previously, we don't want to just test everything. We don't test all of our commensals or non-pathogens, our normal flora. We just want to test the pathogens. When the susceptibility against particular antimicrobials is uncertain, it's not performed on bacteria that's known to be predictably susceptible to common antimicrobial agents or those commonly used to treat infections. For example, group A strep is considered universally susceptible to penicillin, so we wouldn't do antibiotic susceptibility testing unless the patient is on penicillin and it's not working, or if the patient is allergic to penicillin. We may need to do antibiotic susceptibility testing. So factors to consider in susceptibility testing: the body site from which the organism was isolated, the presence of other organisms, so whether we have a pure culture or if it's mixed flora, the quality of the specimen from where the organism was grown, and the host status. So the body site, susceptibility tests are not routinely performed on organisms isolated from an anatomical site for which they are normal inhabitants. For example, E. coli is normally in the GI tract, therefore we're not going to test it when isolated from a stool. Testing of normal biota isolates or isolates likely to represent contamination or colonization should be avoided. So doing so may encourage a physician to treat a non-pathogen and refrain from further investigation of the true cause of the patient's problem. The presence of other bacteria and the quality of the specimen. Pure cultures generally indicate good quality specimens. More than 2 species with greater than 10 to the 5 colony-forming units suggests contamination, especially in urines. Helps in the examination in specimens often contaminated, so sputum and urine. The host status influences susceptibility testing decisions. Normal flora may represent an infection in immunocompromised patients. So we need to know if the patient's immunocompromised, because the things that would make a normal, healthy individual just fine may actually cause infection in someone that's immunocompromised, and may need bactericidal concentrations. Allergies. Patients may be allergic to penicillin, need alternative drug for treatment, and helps make correct antibiotic choice. Selecting antimicrobial agents for testing and reporting. Input and decision should come from laboratory scientists, pharmacists, physicians, and therapeutics committee. There's more than 50 antimicrobials available for treatment. There's plate size constraints, so many antibiotics can fit on the testing panel. So we want to choose commonly used antibiotics. Generally, there's test batteries, and test batteries are kind of combinations of different types of antibiotics that we would test for certain microorganisms. Generally, test batteries are divided into two main groups. There's the gram-positive battery and the gram-negative battery. The gram-positive battery tests for antibiotics that would be useful in treating staphylococci, enterococci, and other types of gram-positive infections. The gram-negative battery would have antibiotics that are typically used for treating infections with enterics, or those gram-negative bacteria that live in the gut, and Pseudomonas. Supplemental battery may be performed in selected cases. So there's urinary tract infections, there may be a urinary battery. So it's just a series of drugs that are typically used to treat urinary tract infections. There's also combination panels that exist. So some panels not only have the ability to do minimum inhibitory concentrations or susceptibilities of different antimicrobials, but it can also give you an identification of the organism. Reporting of susceptibility test results. You can't report susceptibility until you know what the identification of the isolate is. If the isolate ends up not being what you thought it was and you report out an antibiotic susceptibility, you may have to call back those results. Tenets of antimicrobial therapy are to use antibiotics that are the least toxic, the most cost-effective, most clinically approved agent, while avoiding more costly broad-spectrum agents. Broad-spectrum agents cause resistance, so we want to prevent resistance. Note, physician compliance is often difficult. So a lot of times physicians think, or through their training, they think they know which antibiotic that they're wanting to give. Some physicians just give the same antibiotic to every patient regardless of anything. So sometimes even with the information, giving them the information, it's difficult to tell the doctor not to choose certain drugs. Reporting of susceptibility test results. So we don't always want to report out everything, but if we do report out everything, we classify it into three main groups, A, B, and C. We want to report the narrow drugs first before listing secondary drugs, and that helps prevent confusion and overprescription. So Group A antibiotics, report those first. Group B antibiotics, report them if the isolate is resistant to the primary agents, if the patient cannot tolerate the primary agent, the patient has not responded to the primary agent, or the secondary agent would be a better clinical choice for a particular infection or is useful for treating two infections. An example, the primary cephalosporin, acetylzolin, reasonable for E. coli, second or third generation antibiotics are generally not required. Exceptions would be in meningitis. Third generation drugs are better at crossing the blood-brain barrier. The secondary agent should be reported if it's a polymicrobial infection. Polymicrobial infection. Poly, many, microbial, microbes. So many different microbes. Because it might be more effective against all the pathogens. A secondary agent may be reported if the patient has a disseminated infection. Disseminated infection means that it has spread to another area of the body. So it started in one spot and now it has disseminated or spread to another area of the body. Group C, agents with very broad spectrum activity or increased potency, reported for the same reasons noted for Group B, but considered for routine testing if particular institution encounters large numbers of isolates that are resistant to Groups A and B antibiotics. Inoculum preparation and use of McFarland standards. When we are going to inoculate a plate or inoculate a test system for antibiotic susceptibility testing, we need to make sure of the concentration of cells that we're plating. The reason for that is because if we plate too many cells, it can look like they're resistant. If we don't plate enough, it can look like they're susceptible even though they may be resistant. So we want to plate an exact and uniform amount regardless of what we're doing. So in order to prepare an inoculum, there's a couple different methods that we can choose. In method 1 here, it says add cells from 4 to 5 isolated colonies from non-inhibitory agar of similar morphology to a broth medium and allow to grow to log phase. So you isolate some colonies on a plate, you pick 4 to 5 that look the same, you put them into a broth, you stir it up, and then you allow it to grow to log phase, so 18 to 24 hours. You want to sample different colonies, prevent getting mutant colonies or select mutants to get correct susceptibility. A second method is preferred for bacteria that can't grow in broth. So in this method, you would suspend colonies grown overnight on an agar plate directly into the broth or saline. Because this method does not rely on growth in inoculum broth, the use of fresh 16 to 24-hour colonies is imperative. So with this first step, you're isolating colonies, then you're growing them, then you're using them. In this step, you're isolating colonies and using them directly. So this step does cut out some time. The McFarland turbidity standards. So McFarland standards is a series of tubes that have different turbidities or different levels of cloudiness. And the levels of cloudiness has known concentrations of cloudiness in it, or known concentrations of what we would refer to as bacterial cells. So what we can do is we can mix bacteria into tubes and compare it to the standard set of tubes that has been pre-mixed with predetermined concentrations. Then we look at our tube, compare it to the known tube to try to estimate what our concentration is or what our McFarland is. So the number of bacteria to be tested needs to be standardized. Again, I said that too few bacteria looks like it's falsely susceptible. So it looks like the antibiotic's doing a really good job at killing it when really there's just not a lot of bacteria there to begin with. Too many bacteria, you've got safety in numbers. So you've got too clumpy of bacteria, we can see some false resistance happening. Turbidity of the resulting inoculum preparation is compared to that of a known McFarland standard. The turbidity should match prior to performing testing. So I just want to show you, this is a McFarland standard here, and you can see the black line through it. And here is our test sample. You can see that this test sample is too cloudy, so we would have to dilute that until the cloudiness matches this. And we have different McFarland standards. We've got a 1, 2, 3, 0.5, 0.25, 0.75. We've got all different in a row. And then we would shake up our broth culture and compare it to the McFarland standard. And we would want to get to the same level of cloudiness by diluting this down. So standards are specific volumes of 1% sulfuric acid and 1.175% barium chloride. It results in a barium sulfate solution with a specific optical density or turbidity. So inside this McFarland tube here, that's what the chemicals are. It's sulfuric acid and barium chloride. And the cloudiness that's produced is through a double displacement reaction producing barium sulfate. The most common McFarland turbidity standard is 0.5. It contains 99.5 mils of the 1% sulfuric acid and 0.5 mils of the 1.175% barium chloride. Turbidity in the standard is comparable to that of bacterial suspension containing approximately 1.5 times 10 to the 8 CFU per mil. CFU, colony forming units, in other words, cells. So one cell is able to produce a colony, so that's what we refer to as CFUs, colony forming units. You want to vortex the McFarland standard and inoculated broth. Position the tubes side by side under adequate light. Use a white card containing black lines. Look through the broth to the back lines of the card. Add inoculum or diluent as needed. And then there's a nephelometric or spectrophotometric device that can also measure turbidity. So we already looked at this picture, but you can also use one of these devices in order to determine turbidity. So you can get the turbidity of that, the turbidity of this, and see if you have to adjust it. In the clinical setting, it's very unlikely that the clinical setting is going to allow you to use your best judgment and look at it just as a cursory look like this. More often than not, your clinical site is going to have some type of equipment like this to make sure that you are diluting it appropriately. Dilution susceptibility testing methods. So, dilution susceptibility testing methods are designed to determine the minimum inhibitory concentration, or the lowest concentration of the antimicrobial agent required to inhibit the growth of a bacterial isolate. So in other words, you're going to take the antibiotic, you're going to dilute it, and then you're going to test the bacterium in the presence of each one of those diluted concentrations and see what is the minimum concentration that we can go to before the bacteria are no longer impacted or no longer susceptible to that antibiotic. Twofold dilution concentrations are tested, and it's expressed in micrometers or micrograms per mil. The value of the MIC value obtained is compared to known values for resistance. There's 4 results that are possible. Non-susceptible is used when there are no intermediate or resistant interpretive criteria. So if Clinical Laboratory Standards Institute has not developed a breakpoint for what's considered intermediate and what's considered resistance, they don't exist. We can either be susceptible or non-susceptible. We couldn't be susceptible or resistant or susceptible or intermediate. We would have to be susceptible or non-susceptible. So non-susceptible is used only in those cases. Otherwise, for everything else that has established values or established reference values, we would use susceptible, intermediate, or resistant. Susceptible means that the bacteria is susceptible to the antibiotic. In other words, the antibiotic kills the bacteria. Intermediate is an intermediate range where some bacteria may die, others may not, so that's kind of an intermediate range. And resistant means that that bacteria resists the antibiotic. So the antibiotic does not kill the bacteria. The bacteria grows in the presence of the antibiotic. Here's a table showing some standard results. So when looking at this, you would look at the zone of inhibition. You'd look at clearing around the growth. So here you can see susceptible would be less than 8 millimeters, intermediate would be 16, and greater than 32 would be considered resistant. Broth microdilution tests. So after an overnight incubation at 35 degrees Celsius, the minimum inhibitory concentration is determined. It's demonstrated by the absence of turbidity. The final concentration of the inoculum suspension at the time of transfer is 5 times 10 to the 5th CFU per mil. The MIC for each antibiotic tested is the lowest concentration showing no growth, noted as turbidity, haze, or pellet at the bottom of the well. So this is what those dilution trays look like. It's typically 96 well plate. And then these are broth microdilution test equipment. So we would put the bacteria in each one of these wells, and then this magnifies each one of those wells so that we can manually read it. And then this is a very old version of the equipment that we have in the lab, this microscan. The microscan is an automated instrument that will read the levels of turbidity in each one of those wells for us and then report out what's the MIC for each different antibiotic that has been tested. The breakpoint or cutoff for MIC panels is only one or a few concentrations of each antimicrobial agent are tested in a single panel. So the breakpoint is the concentration of the antimicrobial agent that coincides with a susceptible or intermediate MIC breakpoint for a particular drug. It's based on 2 wells. The last well with no growth, or the last well of susceptibility, when decreasing the concentration of the antibiotic. So this is like a fake plate, but if you see, we start with the different tested components, and we put the bacteria in this plate. So for antibiotic A, we're going to have the highest concentration here, and we're decreasing the concentration as we go down in number here, or up in number. So this is the highest concentration, this is a lower concentration, lower, 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 down to no concentration at all. So in the absence of any bacteria, or any antibiotic, all bacteria should grow. But no growth is in the highest concentration of the drug. So you can see there's no growth, and then eventually you see no growth, no growth, no growth, no growth, no growth. That means that the drug is killing the bacteria, but then you get to this point and you see these two wells here. There's no growth here and there's growth here. So the bacteria has been able, or the antibiotic has been able to kill the bacteria for all of these smaller, smaller, smaller concentrations, but this is the break point. At the next smallest concentration, the bacteria are able to grow. So we see minimum inhibitory concentrations with the ones that are labeled with an X. That's the last well where the antibiotic is still working to kill the microorganism before the microorganism overcomes that antibiotic, or before the concentration of the antibiotic decreases so much that it's no longer impactful. Agar dilution tests are a little different. So in this case, we make agar plates with antibiotics at different concentrations. So one will have penicillin at 1 microgram. The other one will have penicillin at 2 microgram. The other one's penicillin at 3 micrograms. So we have these plates with all these different concentrations of antibiotic. Then we put multiple different bacterial colonies on those plates. So as many as 32 different isolates can be simultaneously inoculated onto each 100 millimeter round petri dish of enhanced media. The minimum inhibitory concentration is read as the lowest concentration of antimicrobial agent that inhibits visible growth. So we would look at the plates and where the bacteria is growing or not growing on the plate. We would track that colony and what it looks like on the 1 plate, on the 2 plate, on the 3 plate, on the 4 plate, and see where did it start growing again. Or where did it stop growing? The limitations, there's a short shelf life. So once you create those plates, and it's really time consuming to create the plates, they only last for a week. They must be stored in the refrigerator. It is labor intensive, but it's still used in gonorrhea and anaerobes as the reference method. Disc diffusion testing. So hopefully you covered this in your general microbiology course, but we are going to go through some of the steps of the Kirby-Bauer test. So you swab a 0.5 McFarland standard suspension of bacteria over the entire surface of an agar plate. You want to make a lawn of growth. Add a paper disc containing antibiotic concentrations and then incubate the plate overnight. You're going to then read the diameter of the zone of inhibition and compare to establish zone sizes. Edge of the zone is actually at the minimum inhibitory concentration. You read in millimeters and checked against the standard table and report as non-susceptible, susceptible, intermediate, or resistant. Must be using FDA-approved discs. Long-term storage, store at -20 or below. Refrigerate temperatures for 1 week. Use tightly sealed container with desiccant and bring the disc to room temperature before use to prevent condensation. This is really important, especially when using discs that aren't used often in the clinical lab. You want to make sure that the discs you're putting on the plate are at room temperature, but you don't want them to be at room temperature for too long. So if you're not using them constantly, you got to make sure to return them either to the fridge or freezer, wherever you got them from. So this is a dispenser containing multiple antibiotics. And this is an example of disc diffusion method. So then to get the results from this, so each one of this is one species of bacteria. Each one of these is a different disc that has dropped on the plate. And then you see the zone of inhibition or the zone of clearing around that disc. So you would take the diameter, you'd measure from one side through the center to the other side to determine against the table of what's susceptible, resistant, or intermediate. And then here's using a micrometer to measure the zone of inhibition. There's some very specific details that have to go into making this standardized because we want the same system to be used in every country. So if we establish a protocol, we want to make sure that this protocol is standardized across every country, every lab, everywhere in the world that would use this protocol so that we don't make mistakes and we can get standardized results. That way, if there is an emerging pathogen that's found and it becomes a public health concern that might spread to other countries, we can all share the same data about what we're dealing with. So media formulation, you want to use Miller-Hinton agar. Lawn of growth should be completed. You want to have complete growth throughout the entire plate. Proteus species, you want to ignore the swarming and look at the zone size. So this is not acceptable. Seeing individual colonies like this would not be an acceptable plate to read. We would request a repeat of the test. And then here's the interpretive standards. This is just a sample table of what you would look at. So if I have an ampicillin disc on a plate of staph and I get a reading of 32 millimeters, I would go to ampicillin, staph, I would look at 32, so it would be considered susceptible because 32 is greater than 29. This is a table of primary variables to be controlled for. So this is a really good summary table of the details of how to accurately perform the Kirby-Bauer test. Since hopefully this was covered in your general microbiology course, you're not going to be responsible for memorizing all of these details, but this is just a review and a refresher. So if you do the Kirby-Bauer in your clinical site, you'd be able to remember these details. These details will be on your qualifying exam. They will ask you about agar thickness and incubation temperature, time, and all those types of details. So make sure that you review this before taking your qualifying exam. The E-test. So the E-test, the establishment of an antimicrobial density gradient in an agar medium is a means for determining antimicrobial susceptibility. So plastic test strips are impregnated on the undersurface with an antimicrobial concentration gradient and marked on the upper side with a concentration index or scale. Strips are placed in a radial fashion on the surface of an agar plate that has been inoculated using the disc diffusion preparation technique, so the 0.5 McFarland standard. You incubate them overnight and then look at the plates from the top side with the lids removed. The antimicrobial gradient that forms in the agar around the E-test strip gives rise to elliptical inhibitory areas within each strip. The MIC is determined where the growth ellipse intersects the E-test strip. Other uses for testing fastidious organisms, S. pneumo, H. influenzae, other strep, anaerobic bacteria, even yeasts. So here you can see what the test strip looks like. There's decreasing concentrations of the antibiotic as you go down the test strip. So you can see zone of inhibition is really big and then it kind of gets smaller, 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 smaller. Eventually there is no zone of inhibition anymore. So these, at this concentration, it has no impact on the growth of the microorganism. So we can actually read our minimum inhibitory concentration by looking at where this line intersects with this scale. And then there is a conversion chart that's provided with the manufacturer that allows us to compare this to the minimum inhibitory concentration. So we would take this reading of 0.75 or whatever that is and compare it to the manual that was provided with the E-test strip. Each test strip is a little bit different, so you have to read those manufacturer's guides to determine what the minimum inhibitory concentration is. Modified methods for testing slow-growing or fastidious bacteria. Not all bacteria like to grow on Mueller-Hinton agar. So what do we do if an bacterium doesn't want to grow on that agar? So S. pneumo and strep species don't grow well on the standard medium, so you have to supplement it with 2 to 5% lysed horse blood or 5% sheep's blood. Penicillin is still widely effective, however, increase in resistance due to altered penicillin binding sites means that we might actually have to start testing these guys for resistance or antibiotic susceptibility. You run susceptibility testing on a plate with oxacillin discs, so S. pneumo Greater than 20 millimeters isolate can be safely reported as penicillin susceptible. If it's less than 19, then a penicillin MIC test must be performed. There's also the D-zone test used when isolates appear to be erythromycin resistant and clindamycin sensitive. The purpose is to verify the clindamycin result. So a little bit more about the clindamycin resistance. You put the erythromycin disc next to the clindamycin disc. Erythromycin resistant isolate that appears clindamycin susceptible, you would see the flattening of the clindamycin zone between the two discs. That means that it's inducible clindamycin resistance. So in the presence of erythromycin, it becomes resistant to clindamycin. So this is what the D-zone testing looks like. So you have erythromycin, clindamycin. Clindamycin should be a perfect circle. But because it's resistant to erythromycin, it's inducing resistance of clindamycin. So it's cutting the susceptibility edge short, making this D-shaped image here. So this would mean that we have inducible clindamycin resistance. So under normal circumstances, like if this guy was here and we still had a perfect circle, then it would be truly clindamycin susceptible. But because erythromycin's here, it's causing an induced resistance of clindamycin, and that's what's causing this zone of inhibition to be decreased. H. influenzae and parainfluenzae, so Haemophilus test medium, Miller-Hinton agar base that's supplemented with hemin and NAD, X factor and V factor. Broth and agar forms, you want to incubate in 5 to 7% CO2. Beta-lactamase producers, 20 to 50%, can use ampicillin if not a beta-lactamase producing organism. Gonorrhea, Neisseria gonorrhoeae. Testing is rare. We don't like to handle gonorrhea a lot in the lab, which is probably obvious, but we really don't. We do a lot of molecular testing with gonorrhea, and if we do culture and grow up gonorrhea, the agar base would be used, the GC agar base with 5 to 7% CO2 incubation. Use agar dilution since sometimes it exhibits autolysis. Leads to false susceptibility results. So due to increased penicillin resistance, treatment is shifting to ceftriaxone or a cefizime. Beta-lactamase activity, penicillinase producing Neisseria gonorrhoeae. If altered penicillin binding proteins, then chromosomally mediated resistant and gonorrhoeae. So gonorrhea, you used to just pop some penicillin and get over it. Now there's a lot of resistance out there, a lot of resistant forms. So we are starting to use alternative drugs to treat gonorrhea. And meningitis causes, it's Neisseria meningitidis, which just causes meningitis, a very severe form of bacterial meningitis. Penicillin is the drug of choice, but may be developing resistance. You follow the procedures for streptococci in a carbon dioxide environment. H. pylori and Campylobacter. Agar dilution testing is performed. Mueller-Hinton agar with 5% aged sheep blood. Microaerobic environment, and read at 3 days of incubation. Anaerobes are tested via supplemented Brucella-laked sheep blood agar. Broth microdilution method is commonly used. You incubate in anaerobic conditions for 48 hours, and you can use an E-test. Infrequently encountered or fastidious bacteria. There's a list of bacteria that CLSI gives us guidance for, but these are not frequently encountered. We would usually reference protocols if any of these are encountered in the clinical lab. Additional organisms and testing concerns. So methicillin-resistant organisms. Methicillin, also known as oxacillin resistance, is due to the MEK-A gene, penicillin binding protein with low affinity for all beta-lactam drugs. Methicillin-resistant Staphylococcus aureus is the one of highest concern here. There's also heteroresistance, where all cells in the population have the gene, but not all of them are actively expressing the gene at a given time. So for MRSA testing, we would use Miller-Hinton agar with 4% sodium chloride, and 6 micrometer per mil oxacillin, known as oxacillin screen plate. So you would get a 0.5 McFarland standard, you swab it and make a lawn, incubate it, and if you see more than one colony growing, then we know that we have MRSA. There's a disc diffusion method is better using cephozidin discs, which is easier to interpret. So you throw a cephozidin disc on the plate and see if there's a zone of inhibition or not. This is now considered to be the preferred method versus the other plate method. MRSA are also resistant to penicillins and many other drugs like clindamycin, tetracyclines, sulfamethoxazole, erythromycin, etc. So you report as resistant no matter what the results show. Chromogenic agar is also commonly used. So MRSA on chromogenic agar produces these blue colonies, these denim blue colonies. So these would not be MRSA. The blue ones would be MRSA. So if you do a MRSA plate, you swab it on this plate, and you see the denim blue colonies, you would report this result. This is a very important and critical result that gets called to the treating physician or nurse. So you make the phone call and you do not stop calling until they are notified and you have confirmation. And then in the report, you always want to write who you called, like their name, first and last name, where they worked, what floor they were on, or what their details were, what phone number you called, who you are, the date and time of the call. So you want to document everything about the call that's made to report critical results. Vancomycin resistance. So vancomycin intermediate Staph aureus has reduced susceptibility to vancomycin. Why is this important? Well, if you have MRSA, methicillin resistant Staphylococcus aureus, usually you're given vancomycin to treat that. Well, if vancomycin doesn't work either, you're out of options. There's really nothing else that they can do. So there's other drugs that they might be able to try. There's experimental drugs, but having both of those resistance mechanisms is really dangerous. So that's something that definitely has to be reported. There have been a small number of cases reported in the United States of MRSA and methicillin and vancomycin resistant Staphylococcus aureus, but that's something that's very dangerous. So vancomycin intermediate Staph aureus has a reduced susceptibility. Vancomycin resistant Staph aureus is of great concern because vancomycin has traditionally been a treatment of choice. And then there's population analysis profiles, which VISA and VERSA are reported. Enterococci, for uncomplicated infections, you use ampicillin and penicillin. They're usually effective. But for serious enterococcal infections, such as endocarditis, ampicillin and penicillin typically only effective in combination with an aminoglycoside drug, such as gentamicin, or in some cases, streptomycin. Detection of VRE, so vancomycin-resistant enterococci. This is different from vancomycin-resistant Staphylococcus aureus. So VRE incidence is on the rise. If a patient has VRE, the vancomycin-resistant enterococcus has the ability to give that resistance to the Staphylococcus aureus. So if they already have MRSA, then they get VRE, the likelihood of them getting that gene transfer from the enterococcus to the Staphylococcus is high. So vancomycin screening agar plate, it contains brain heart infusion agar supplemented with vancomycin, and if anything grows on there, if you see more than 2 colonies again, then you know that you have the vancomycin resistant enterococci. There's also a chromogenic agar for VRE. Extended spectrum beta-lactamases, Klebsiella pneumoniae, Oxytoca, and many E. coli are resistant to ampicillin because of the production of plasmid-mediated beta-lactamases. ESBLs, typically susceptible to later generation cephalosporins or azotrinum. Some spontaneous mutations may result in novel beta-lactamases that can inactivate extended spectrum antibiotics. So this is a picture of an ESBL confirmatory test. So it's extended spectrum beta-lactamases, but also you can detect this using some of the automated systems. Some of those microdilution plates would be able to detect whether you have an ESBL. Carbapinamases identified in K. pneumoniae and now in other Enterobacteriaceae. New test, carbapinum inactivation method. So a carbapinum disc is incubated in water or tryptic soy broth containing a suspension of bacteria to be tested for 4 hours. Inactivation of the disc is analyzed by placing the carbapinum disc on a lawn plate of carbapinum susceptible control strain. Results in no zone of inhibition after an overnight incubation. Automated antimicrobial susceptibility test methods. So the principles of the technologies used, different levels of automation. One instrument interprets growth endpoints of broth microdilution panels only when placed on an automatic reader device. Other instruments are considered hands-off. 2 optical approaches, so turbidimetric, using a photometer to examine test wells, and the detection of hydrolysis of a fluorogenic growth substrate incorporated in a special test medium. It's detected by a fluorometer as emission of a fluorescent signal. All devices offer a view box or mirror to facilitate manual reading of the results after incubation. Many of the common automated instrument systems are listed below: the BD Phoenix system, Microscan Walkaway, Trek, Sensitor, and the Vitek 1.2 and Vitek 2 Compact. The ones that I've more often seen in practice are the Microscan and the Vitek. The one we have in the lab is the Microscan, but we don't have the Walkaway, we just have the simple reader. So this is the Walkaway system. So with the Walkaway system, you're able to put the plate in there and it incubates it and reads it at the exact time that's necessary. The system that we have in the lab is this system. So we would incubate it in a separate incubator and then we would manually transfer it here when it's ready to be read. And then it'll read it for us. This is the inoculator device that we use, the Renoc device. So this allows us to inoculate these well plates so that all of the different concentrations can be read by the system. There is a supplemental video provided that will talk about the Renoc inoculator device. The Vitek. So in this card, there's different concentrations of different types of antibiotics, and there's a barcode. So you inoculate this with all of the different, with the one species of isolated bacteria. So the McFarland standard that you've created gets inoculated on the card gets put into the system, and then the computer system will identify whether there's growth or not in each one of the wells. Methods of detecting antimicrobial inactivating enzymes. So beta-lactamase enzymes chemically inactivate beta-lactam molecules by disrupting the beta-lactam ring component of the molecule. Production of beta-lactamase is a significant mechanism contributing to resistance. The most common test uses the chromogenic cephalosporin nitrophin. Cephinase discs consist of filter paper impregnated with nitrocephin. The disc is moistened with water. A loop full of organism is applied to the disc, and then you read results after the proper wait time. If the bacteria produces beta-lactamase, you're going to see a pink or red color develop. If there's no color change, it's negative. So here you can see that disc. You smear the bacteria on the disc, and if it's pink, it's positive. If it's yellow, it's negative.